Growth is a basic property of life. All living things grow, plants and animals. Human beings grow. But what is the biological basis of growth? The living cell of any species holds the answer to this question. The simplest answer to why living things grow is that most cells divide through a process called mitosis. Through this division, two descendant cells are produced from one. Mitosis is by no means a simple process. The cell accomplishes two basic functions in the process of mitosis. We'll illustrate these two functions, beginning with this model of a single cell. First, all the organelles, or structures of the cytoplasm of the original cell, will be divided among two descendant cells. Second, the all-important hereditary material in the nucleus of the cell will be duplicated so that each of the descendant cells contains the same amount of hereditary material. This duplication stage of mitosis begins within the chromosomes of the nucleus. In this photomicrograph of a cell, the chromosomes are the dark, coiled mass. Here are unusually large chromosomes seen under higher magnification. The chromosomes contain genes, which are in turn made up of molecules of a submicroscopic chemical compound called DNA. This is a molecular model of DNA. The DNA, or deoxyribose nucleic acid molecule, is an arrangement of smaller molecules called nucleotides. The particular arrangement of nucleotides within the DNA molecule determines the particular species of a living thing and differences between individuals of the same species. Each DNA nucleotide consists of three chemical components. The first molecular component of the nucleotide is always phosphoric acid, a compound of phosphorus. Linked to this is the second molecular component, always deoxyribose, a simple sugar. Linked to deoxyribose is a third molecular component. This is guanine. However, the third component of a DNA nucleotide is not always guanine. The third molecule varies. It may be the chemical compound guanine, or it may be cytosine, adenine, or thymine. These are the only four chemical combinations that produce DNA nucleotides. However, these nucleotides may be arranged in several different orders. Using the four nucleotides, we could devise a maximum of 24 different arrangements. If we were to add duplicates of the four nucleotides, there could be almost no end to the number of arrangements. Much of our knowledge about DNA and its component nucleotides is based on a study in which a wide variety of cells were analyzed with specialized equipment. Dr. Maurice Wilkins was one of the men who guided this study. Dr. James Watson made other contributions to the study, along with Dr. Francis Crick. These men were awarded the Nobel Prize in Medicine in 1962. Their model of the DNA molecule indicates that the nucleotides are arranged in a double spiral. To clarify this important concept, let's look at this much simpler model. This ladder is the first step in the shape of the DNA molecule. The uprights represent a linked chain of phosphoric acid and deoxyribose molecules, the two molecules which are always part of the DNA nucleotides. The rungs represent molecules of the third compounds which complete the nucleotides. These are guanine and cytosine, thymine and adenine. The molecules of thymine and adenine are chemically bonded or locked together. In a similar way, molecules of cytosine and guanine are also chemically bonded. Thus, we have an arrangement of many DNA nucleotides linked in a DNA molecule. To produce the proper physical shape, we twist the uprights of the ladder about each other. This gives us the double spiral shape of the DNA molecule. Variations in the structure of DNA account for the thousands of different living species. For the variations which may occur in the arrangement of the nucleotides within the molecule are practically endless. Variations of DNA molecules within the chromosomes of cells determine whether an organism will be a rose, or a chick, or a human being. Variations in DNA molecules also determine the unique characteristics by which one individual differs from other members of the same species. If living things are to grow, the hereditary material of DNA must be duplicated in the cells of the organism. Our next step, then, is to see how the DNA molecule is duplicated in mitosis, or cell reproduction. We'll untwist the model to simplify matters. You'll remember that the rungs of the DNA molecule are held together by chemical bonds. These bonds are very weak and break when the cell is sufficiently mature to reproduce. The DNA molecule becomes unzipped. When all the bonds are broken, two half DNA molecules result. During cell reproduction, each half of the DNA molecule becomes the basis of a complete new DNA molecule. To see how each half of the DNA molecule is duplicated, let's start with one half. From the large variety of chemical compounds stored in the cell, each half receives certain nucleotides. Some nucleotides lock into place, others are rejected. Those nucleotides that lock into place are arranged in the order established in the original DNA molecule. Now the bonds linking the nucleotides are formed, completing a new DNA molecule. This exact duplication of the DNA molecule is the means by which the physical characteristics of a cell are passed on to its descendants. Keep in mind that duplication of DNA occurs within the chromosomes of the cell during the first step of mitosis. The process of mitosis occurs in 5 major phases. It is in the first, or interphase, that duplication of the DNA molecules in the chromosomes takes place. Beginning with the prophase, the second phase of mitosis, the entire process may be viewed as it happens under the microscope. It is sometimes difficult to follow the action in mitosis, so here's what happens in the prophase. The spindle fibers in the cytoplasm form about small bodies called centrioles, which are not seen in certain plant cells. Also watch the chromosomes within the nucleus. The chromosomes, which contain the DNA molecules, appear as long strands. As these strands become shorter and shorter due to a coiling process, we see that each chromosome is double. The double strands are joined at the centromere, a central point on the chromosome. Each half of the original chromosome is now referred to as a chromatid. As each chromatid attains its minimum length and maximum thickness, the nucleoli, small structures within the nucleus, and the nuclear membrane gradually disappear. The centrioles, with their spindle fibers, have moved to positions called poles of the cell on opposite sides of the nucleus. The spindle fibers are now attached to the centromeres of the chromatids. Now, through cinemicroscopy, we see a living cell in the prophase of mitosis. Notice the dark chromosomes coiling and shortening. Note that the chromosomes are double and joined at a dark spot. These are the centromeres. The end of the prophase has been reached. Let's consider now the metaphase, the third phase of mitosis. In the metaphase, the chromosomes line up between opposite poles of the cell. Here's the same action under the microscope. This alignment of the chromosomes completes the metaphase. In the fourth phase of mitosis, the anaphase, the cell prepares for division of the cytoplasm. In the anaphase, the centromeres of the chromatids divide. This produces a pair of chromosomes. Each of the pair moves in opposite directions along the spindle fibers. The anaphase ends when the two identical sets of chromosomes have reached the opposite poles of the cell. Here's the anaphase as it actually occurs. The chromosomes move to opposite poles of the cell. Following the anaphase, the cell enters the telophase, the final stage of mitosis. The actual process of division of the cytoplasm, the second major function of mitosis, takes place in the telophase. In plant cells, the telophase begins with the formation of a cell plate that tends to split the cell in half. In animal cells, this splitting is accomplished through the furrowing of the cell membrane. In some cells, this furrowing action may take place earlier in the anaphase. When this furrowing is completed, two cells are formed. A nuclear membrane appears in each cell. As the spindle fibers gradually disappear, the chromosomes uncoil and lengthen. With the reappearance of the nucleoli and the nuclear membrane and the doubling of the centrioles, the process of mitosis has been completed. Now the telophase as seen under the microscope. The furrowing of the cell membrane begins the actual division of the cell. Two living organisms now exist where there was one. In this rapid motion scene, the entire mitotic process is condensed. Through mitosis, the hereditary material of the cell nucleus is duplicated so that each cell exactly reproduces its own hereditary characteristics. In addition, the cytoplasm divides, thus providing for growth of the organism. Aside from mitosis, certain cells, sex cells, reproduce through a process called meiosis, or reduction division. Meiosis differs from mitosis in that when the sex cells reproduce, only half of the chromosomes present in the parent cell are passed on to each daughter cell. Meiosis occurs in two divisions. These cells are undergoing the first division. These cells are undergoing the second division. Meiosis occurs only in the sex cells and is the process by which the hereditary characteristics of an individual are passed on to its offspring. There are then two types of cell division. Both involve the duplication of DNA. Meiosis is a specialized division among sex cells, making possible the reproduction of a species. Mitosis is a more general division by which living things grow.